everyone, and welcome to today's webinar. I'll just give people a few moments to get settled before we begin. Hope everyone's enjoying this nice summer day. Okay, so I think I will kick things off. So good morning or good afternoon, everyone. My name is Caitlin Jaggers. I am the research lead at the Alzheimer Society of Canada within the Research, Knowledge, Translation, and Exchange team. The Alzheimer Society of Canada is very pleased to welcome you to another ASRP Exchange presentation that is being offered through our provincial support program. This series has been developed to feature Alzheimer Society Research Program recipients who have completed their funding and who will speak to the outcomes of their ASRP-funded research while addressing how their results have and will continue to impact people with lived experience. Joining us today is Daisy Couture from the Alzheimer Society of British Columbia, who will be assisting and moderating today's question and answer period following the presentation. I would like to start off by going through an agenda for today's presentation, and then I will pass it over to our presenter, Dr. Da Ma. Before we begin, please note this webinar is estimated to be under 1 hour in length and is being recorded so that only the video and audio of the presenters is captured during this recording. The presentation recording, in addition to the PowerPoint slides in PDF form, will be made available on Connection for Alzheimer Society staff, as well as on the Alzheimer Society of Canada's website on our ASRP Exchange webpage. Today's webinar will be presented by Dr. Da Ma and is being offered as one of our ASRP Exchange webinars within the provincial support program Meet the Researcher series. We are so very pleased to have Dr. Da Ma here with us today to present his project, Eye Imaging in Dementia Models, Understanding the Progression of the Tau Protein. This project was funded by the Alzheimer Society Research Program, starting in September of 2018, with project completion ending in September of 2020. Dr. Da Ma received his master's degree from the University of Hong Kong in the field of biomedical engineering and ophthalmology. He then received his doctoral degree from University College London, where he specialized in medical and biomedical imaging. He is now a postdoctoral research fellow at Simon Fraser University in the School of Engineering Science. Dr. Da Ma's research interest is to study neurodegeneration for both the brain and the retina using medical imaging and computational neuroanatomy. His research work ranges from brain and retinal imaging to translational applications in both clinical and small animal studies. As mentioned, at the end of today's presentation, Daisy Couture will be moderating a short question and answer period. Please use the Zoom chat box feature if you have any comments you would like to make or questions you would like to pose during the presentation. Please note we will hold off on asking our presenter to respond until after he has completed his presentation. Without further ado, please join me in welcoming Dr. Da Ma.
Thank you, Katie, for the kind introductions. Let me share my screens and see whether you can see my screens perfectly. Okay, so before I start my presentation, just to check whether everyone can hear me clearly and see my slides clearly.
Yes, we can hear you and see your slides.
Perfect. Thank you, Katie. So, yeah, just to go through the first slides. So today, I'm honored to give the presentation of my Alzheimer's Research Society Program project, which I entitled In Search of Eye Imaging Biomarkers in Dementia and Mouse Models, Understanding the Role of Tau and Amyloid Beta Proteins in the Retina. So as Kate mentioned, this is an Alzheimer's Society supported project conducted in the Simon Fraser University in collaboration with different institutions like University College London, as well as the University of British Columbia. So just to start with, I think what I'm going to show is here is a big picture about the ASRP project that I'm going to present today. So in the middle is the overarching research goals of the project, which is to study the Alzheimer's related pathologies in the eyes using mouse models. And specifically, if you look at the top rows, we will use different medical imaging techniques to study the presence of two types of the pathological proteins in the retina, which are the clinical indications of onset for Alzheimer's disease. So the first is called the hyperphosphorylated tau proteins, and the second is called the amyloid protein plaques. And I will elaborate more details about the retinal pathologies of these two types of pathologies in more details in the later slides. And in this presentation, I also want to focus on two of other important aspects of the project. The first one is to connecting the retinal pathologies to the brain pathologies, which is important to sort of see whether the actual diagnosis can be achieved in the other organs other than the brains. And the other part is to linking the live imagings from the mouse imaging to the clinical imaging, which is important for clinical translation purposes, and see whether we can actually make the techniques that we built and the knowledge we built and translate that into the clinical practice. So without further ado, I will try to explain the motivations behind this project. And so why are we trying to use retinal imaging for diagnosis of Alzheimer's disease? If we understand biologically, the retina, you might know that the retina is extension of the central nervous system, or the CNS. And as shown in the schematic diagram on the right, you can see there the retina is connected to the brain directly through the optical nerve fibers. And both the retinas and the brains are sharing the same embryonic origins. And also, if you can see the diagrams in the middle, they also share similar anatomical structures, in the sense that they are laminar structures constituted of different different types of the neural cell types, and the signal transport are coming from the directions vertical to the laminars in both the retina and the cortical side. Then also, the retina and the brains are functionally connected. As I mentioned, the visual signals will transfer from the retinas all the way to the back of the brain, which is the visual cortex, through the optical nerves. And finally, one of the important aspect is that the retina is sort of have been considered as the windows of the brains, and compared to the brains, which is protected by the bony skulls, and it's harder to image in the brains, where you need to use imaging techniques like MRI or PET, which is more expensive and providing sort of a image with the lesser resolutions. But comparatively, the retina can be used can be imaged in vivo using some non-invasive light-based imaging techniques, and those techniques can provide much higher resolution images compared to the neural image techniques such as MRI and PET in the brains, and it's also a very cost-effective way to image the neurons. So with the motivation presented, I also want to show the important questions to ask. So does Alzheimer's disease pathologies actually happen or manifested in the eyes in the first place? And if it does, how can we detect it? And if we can, then what stage of the diseases can we detect it? And because the early diagnosis is a crucial part of the Alzheimer's disease. That's why we need to make sure that we can evaluate the pathologic onset at the reasonable time, at the reasonable early stage. And these are the questions that we want to we attempt to answer in these ASRP projects. So as we all know, the Alzheimer's disease is a complex multifactorial disease that manifests in multiple pathologies, including, for example, the vascular disruption, the abnormal protein expressions and aggregations, or the inflammations related to immunoresponses, and eventually leading to the neuronal degenerations. So those are the two hallmarkers that we will be looking at specifically, as I mentioned, the tau and amyloid. And the amyloid is sort of a toxic proteins coming from the amyloid precursor proteins. And also the tau proteins is a toxic form of another proteins which sits inside the neuronal cells called microtubule associated proteins. And the plaques form of the amyloid and a hyperphosphorylated form of the taus will form the toxicities, both external to the neuronal cells as well as internal to the neural cells. And the figures on the right is extracted from a recent review papers and showing a sort of both of those toxic proteins are actually found in the retina, indicating the possibility of using that as a biomarkers. But we also want to find whether they are presented in the case of Alzheimer's and whether those associations of the pathologies and disease have a higher specificity and sensitivity. So the first project I will be presenting is to looking at the tau pathology in the retina, as well as the optic nerves, as well, and also its associations with the brain pathologies. And in this project, we were using transgenic mouse models with tau pathology called RTG4510. And this is in collaboration, close collaboration between the Simon Fraser University as well as the University College London. And to start with, we will first confirm the tau pathologies in the brains of the mouse models first. And we have using the immunohistochemistry stainings for the hyperphosphorylated tau. And the figure on the left is showing the histological slices and of the transgenic mouse models. And the deep dark brown color actually indicating the immunoactivities or the increase of the immunoactivities in the transgenic mouse model compared to the wild type. And the figure on the right is showing the quantitative analysis about the stainings and showing there's indeed a significant increase of the immunoactivities in the transgenic mouse model. So with the confirmation of the brain pathologies, we also want to see whether these kind of the additional protein depositions actually leading to the structural changes, which is an indication of the neurodegeneration. And so we use some automatic quantitative methods to parcelate the brain structures and then evaluate or quantify the brain volumes, structural volumes, with the normalized with the brain, the total brain volumes. And we extracted 4 regions to quantify the neurodegenerations, being the cortex, the hippocampus, the thalamus, and the superior colliculum. The cortex and the hippocampus are the 2 structures which are closely related to the Alzheimer's disease, or mostly affected by Alzheimer's disease. And the quantitative result of the brain structural volumes normalized to the total brain volumes indeed showed a significant reduction of the volume, indicating a certain level of the neurodegeneration. And there is also a significant increase in thalamus, which is an interesting finding from the study. So basically, those are the confirmation from the brain side. And then moving to the backwards along the visual pathway. First, we look at the change of the volumes for the optical nerve itself. So we can use because during the magnetic resonance imaging of the entire brains, we can also image the retinas and optic nerves. So we again segmented and quantified the volumes, and there is indeed showing a significant reduction of the volumes in the transgenic mouse, which is shown in the red compared to the wild type model, which is shown in the blue. And furthermore, another thing we quantified is the signal intensity of the MRI data in the optical nerve. So the MRI data, a little background about MRI data is that we are using the T2 weighted MRI imaging, where the signals indicating the content of the water. So the quantitative result shows there is a significant increase of the signal intensities in the transgenic mouse, as shown in the red compared to the wild type, which is an indication of the increased water content, basically reflecting some kind of edemas happens in the optic nerve, indicating a neurodegeneration or some pathologies derived from the tau pathology. So moving further back to the eyes, we're now looking at the tau depositions in the retina. So this is a representative slide showing the immunohistochemistry staining of the retinal samples. And there's a schematic diagram of the retina on the top. So basically, we are looking from the top to bottoms, and different colors indicating the different types of the neurons stained in different retinal layers. So the green shows the total tau, including both the healthy tau proteins as well as the phosphorylated toxic taus, and the red indicating the specific staining of the toxic phosphorylating taus. And the blue one is the staining of the nucleus, indicating whether there is enough cell number of cells, neuronal cells, or healthy neuronal cells in the retina. So with that in mind, let's look at the results of the immunohistochemistry stainings. On the top rows, it is the stainings of the wild type, and on the bottom rows, it's the staining of the transgenic mouse model. And if you look at on the left columns, both of the mouse groups are showing the a certain number of the total tau proteins deposits in the inner part of the retina, and also the number of the nuclear cells can be shown in the blue in the relatively outer side of the retina, although it's still part of the inner retina. And if we look at the columns on the right, which shows the phosphorylated tau, a significant increase of the levels for the phosphorylated toxic tau can be detected both in the outer retina, which is a photoreceptor layer, as well as the inner retina, which is the ganglion cell layer, as well as nuclear and plexiform layers, indicating a certain level of increase, indeed presented in the postmortem retinal stainings in the slices, in the historical slices. And here's the quantitative results showing the reflecting what we've observed in the immunohistochemistry stainings. And first, we quantified the percentage of the immunostain positive cells and compared to the wild type cell numbers, which is shown in the blues. Significant increase in the transgenic mouse model, as shown in the red, can be seen both in the central retina as well as the peripheral retina in both of the two layers in the inner retina. And if we look at the intensity of the phosphorylated tau stainings, there is also a significant increase in the especially in the peripheral side of the new retina, indicating that some of the tau-related pathologies actually started to happen in the peripheral side of the retina and at the early stage, and then it may be propagated to the central part of the retina. And another important part is to quantify the neurodegeneration in the retina as well, mirroring what we studied in the brains. So to do that, we quantified the two things. One specifically for the nucleus, which is stained in blue. So we quantified the number of nuclear cells or the nuclear cell densities in the retina, or specifically in the nuclear layer of the retina. Also, we quantified the relative thickness of the inner plexiform layers. A thickness can be considered as a later stage of the neurodegenerations. And if we look at the data, the figure on the left shows the nuclear densities in the most inner part of the retina, the retinal ganglion cell layers. And the densities in the transgenic mouse model, as shown in the red, is indeed significantly smaller compared to the wild types, which is shown in blue, both for the peripheral and the central part. And similar thing happened in the inner nuclear layers, and indicating but in a less severe manner, indicating the level of the neurodegenerations are actually layer dependent. And again, when we look at the thickness changes, there is no significant change happened yet in the inner layer of the retina. So that means that there is a certain level of the neurodegeneration in the cellular level. However, it's not manifested into the structural level, so that we can detect it in a more gross manner. So in summary to summarize this part of the project, what we are looking at is this is a schematic diagram showing a significant increase of the tau pathologies in both the retina as well as the brains, as shown in the darker blue side. And also, those are those changes are linked between the retina and the brains are linked through the optic nerve tract. And so I will just take the opportunity to see the connections of the in the small animal studies and compare that to the clinical studies. We did include some of the clinical MRI scans for dementia patients with tau pathologies, and we look at the optical nerve volumes and also see a significant reduction in optical nerve volumes, mirroring what we find in the transgenic mouse models, indicating a level of connections to the clinical practice. And so this is the main project we conducted in the ASRP to look at the tau pathology. I just want to briefly mention about another parallel project we are doing for imaging, in vivo imaging of the amyloid beta pathology, because actually later in the project we found that there is interconnections between the two types of the proteins, and in order to have a fully understanding of the pathologies, we need to understand both of them in a big picture manner. Similar to what we do in the first project, we first do the immunohistochemistry stainings in the mouse models and for the retinas. And here the darker brown immunohistochemistry indicating a higher level of the immunoactivities in the transgenic mouse compared to the wild-type mouse. And if we look at the figures on the right, the quantitative result of the retinal stainings indeed show the significant increase. What is interesting is that in this project, we are doing the longitudinal, or we actually imaging the mouses at different time points and look at their data. Interestingly, the increase of the immunostainings actually is according to the age in the transgenic mouse. However, that's not happened in the wild-type mouse, meaning that there's a age-related degenerations happen in the amyloid beta pathologies. And we trying to develop some in vivo retinal fluorescent imaging techniques to look at to confirm the findings in the ex vivo studies. So we are using the tail vein injected compound called curcumin, which is a natural compound which can bind to the amyloid proteins. And so the image on the left, bottom left, shows the in vivo fluorescent imaging after the injection of the curcumin. And we devised a quantification called fluorescent index from the images. And the figures on the right shows the quantitative result of the fluorescent index. And there is there is no significant difference in the early stage of the transgenic model compared to the wild type. However, in the later stage, there is indeed a significant increase in the transgenic mouse, which becomes significantly higher compared to the wild types, indicating the level of the amyloidopathy has increased. And again, we compared that to the brain studies and to show to have a correlation analysis between the ex vivo cortical staining of the amyloid beta, as shown in the x-axis, and the fluorescent index that we show in the in vivo in the y-axis. There is a significant correlations in the transgenic mouse. However, there's a less significance in the wild types, indicating there might be a certain level of the false positives in the metrics that we developed, so that we have to fine-tune the metrics better in a later stage. And in a parallel study, there is we did some correlation from to connect the studies or the findings between the transgenic animal models to the human studies. So here is a slide, representative slide showing the images of the immunohistochemistry staining for the post-mortem human retinas. And in the bottom rows, we can see there is an increased level of the amyloid beta immunohistochemistry stainings as shown in red compared to the blue. And also the level of the nuclear cells is decreased compared to the control group of healthy group as shown in the top. So that's also showing the connections between the clinical result in the human retinas and compared to or connected to the small animal studies in the transgenic mouse model. So just to say something about moving forward these projects, we are looking at some single modalities of retinal imaging. As I said, if we want to have a big picture about the pathological cascade in the mouse models or in the retinas for the Alzheimer's disease, we want to integrate multi-model imaging modalities. So we are building some optical-based retinal imaging modalities to look at both the retinal structural change as well as the vascular change. And so here is showing the sort of most updated Alzheimer's pathological models. In these models, as shown on the right, you can see there is a vascular factors happening at the first stage at the initial stage of Alzheimer's, and then at the second stage of Alzheimer's, the amyloid beta and the tau protein aggregation happens, and eventually leading to the neurodegeneration. So actually including the vascular and the structural informations is important to give a more big picture. So here is some of the in vivo retinal imaging techniques that we have been building in the group, mainly using the optical coherence tomography to look at the structural and the angiography of the retinas, and as well as using 2-photon exciting fluorescent imagings to look at the retinal angiography as well as even the fluorescent labeled single cells. And those can be combined together with the techniques. And finally, just want to say quantification is important. So along with the development of the multimodal retinal imaging, we are also developing the quantitative morphological analysis method on the OCT structural images to look at the different layer changes and potentially looking at the vascular change when applying that to the vascular imaging as well. So with that said, I want to just say thanks to the project, to acknowledge all the collaborators I resided in the Simon Fraser University, the University of British Columbia, as well as the University of College London. And I just want to say that this is both of the projects are highly collaborative, and a lot of the members are helping with the project. And I can't include everyone that helps, just including most of the primary lab members that helps to push forward this project. And with that said, I will conclude my presentations, and thank you for listening. I'm very welcome to all the questions and looking forward to the discussion sessions.
Thank you, Das. So if you don't mind just unsharing your screen, and I'll pull my slides back up.
Sure.
And we will, as mentioned, engage in a question and answer period now. So we'll open up the floor to our audience. If anyone has any questions or comments you'd like to ask our presenter, you can post those in the chat box or the Q&A box, and we will we'll share those with the presenter, and he will answer to the best of his abilities according to his research and his experience. But while we wait for any questions or comments to come in, I just want to say that that was a very interesting presentation. I certainly learned a lot. It's actually not something I'm overly familiar with. The retinal side of dementia is quite intriguing to learn more about how that can be utilized and really taking that to another level. I was definitely very, very interested in your presentation and learned a lot. So thank you for taking the time to pull that together for us today.
Thank you, Katie. Yeah, just want to say it's an interesting project for me as well. It's combining different domains from the images and looking at some of the state of the art in the realm of Alzheimer's disease. And yes, hope we can find some alternative solutions to provide with the current diagnosis methods.
Yeah, I think that was so interesting. I just wanted to ask a quick question myself if we're waiting for anything, is about that exactly. Is, of course, I know that most of your work was done in mouse models, of course, but thinking into the future, is it your hope that there would be, like, what do you think it would actually look like if this manages to be effective and translated into humans? Do you think it would be, you know, a one scan that you might get at when you go to an optometrist visit, or would it be actually many different modes of scans that you'd be getting, you know, if you are starting to have concerns and have seen a neurologist?
Yeah, thank you, Daisy. That's a fantastic question. This is actually I think some of the questions or the difficulties we have to address in the preclinical studies where we're using the animal models for all kinds of the preclinical studies. So specifically in the retinal imaging. So the first stage we are looking at, as I presented, is to confirm the presence of pathology first. So we are using different post-mortem analysis to just make sure, because in the literature, there is a lot of discrepancies between whether, how or when we can detect those pathologies and how severe they are. And after that, we need to define whether we can image that in vivo, right? So that we don't need to, because we have to, if we want to image them in humans, we have to make sure that it's in the live mouse system can work. And so that's what our in vivo imaging comes in for the specific for amyloid beta. And what's the difficult is that even for the in vivo imaging, if we are using fluorescence, we need to have some dyes injected. So for the amyloid beta, now we are so a lot of the other groups that are presented in the slides actually are using some oral intake of the curcumin, which avoids the injections to make it less invasive. However, just as I mentioned, as a thing, disease like Alzheimer's disease are multifactorial. So just by looking at, as you mentioned, just by looking at one pathology might not be enough to differentiate to other diseases, as both amyloid beta and tau are presented in different kind of diseases in the retina. So multimodal is the way to go. That's why we want to incorporate both the optical coherent tomography and fluorescent imaging and all other types of imagings, and to make it less invasive, no injection needed, and multiple informations can be included. structural vasculature. And what we also are looking at now is the dynamic contrast imaging, which is something that we want to utilize. Basically, if we can image the patient in a short period of time with multiple time points, then we can actually calculate the dynamic changes in the molecular level or in the cellular level. And then those are the informations that might give us hints about the specificity of the pathologies to differentiate Alzheimer's to others. And yeah, hopefully, yeah, so those are the things that we are still currently actively working on.
Oh, thank you. That's so interesting. And I mean, I guess it makes a lot of sense. You know, Alzheimer's is such a complicated disease in the brain. Why would it be any simpler in the eye, if not more complicated? But I think that's so fascinating about those challenges that I wouldn't have thought of before, especially with the injections versus oral supplements to make it actually non-invasive. And on that line, we have a question from Monica, who's following up about that, who's curious about, and of course this might be speculative, but about how far away you think, you know, a version of this might be appearing in a clinical setting, and what needs to happen before it can actually start to be used in diagnosis.
Right. I think those are the two important questions. First, how far away we can actually use it. Actually, I think there are the reason we focus on the amyloid beta in the second stage is that it's closer to the clinical practice, because we found the compound, which is the field found the compound amyloid beta, which we also take in our studies, and which is safe because it's natural compound, and that's why we can actually make it easier to the clinical stage, as even to the stage of being approved. And while for the tau, the problem is that we still need to find the compounds if we want to look at fluorescent imaging that is less invasive, or and also so those are the two closest two imaging modalities that are closer to the clinical translations, because it's readily available. While the optical coherence tomography itself, the image modalities I mentioned at the last stage, is still in active development, and we still need to find, because we need to find the clinical relevant informations. Even we find the dynamic contrast, which are highly correlated to the Alzheimer's disease, we need to have basic science research to understand what those contrast means in terms of the molecular mechanisms, in order to make it to the clinics and to approve. So that's still in the early stage of the research, but we have a higher hope that it can be pushed forward and eventually become clinically translatable.
Interesting. That's, yeah, I guess with the multimodal things, things are at different levels of development, and they all show different challenges, which all you have to meet to make them actually work clinically.
Yes, exactly.
We have a question from Bernice here, who was wondering if there's any correlation between, she's specifically asking about thin retinas and the risk of developing Alzheimer's disease. So I guess that's an idea about, is there something structurally about the eye that we, could see in advance of pathology?
Right. I think the question is specifically addressing the retinal thickness.
Yeah.
So it has been shown for a long time that there's a correlation between the retinal thinning and the Alzheimer's disease, even at the quite early stage, not as early as other pathology can show. However, the problem of the retinal thickness is that it's an indication of the either neurodegeneration or the in a broad sense. So just by looking at the thickness itself, we cannot say it can have enough specificity to indicating Alzheimer's disease. We can only say that either it's risk factors or it's a consequence of the neurodegeneration. But by itself, I don't think we need to just having the thinner retina itself. I don't think we have to worry about it had definitive concern about Alzheimer's. And also we have to look at the individual variabilities. So the thinner, it's a change of the retinal thickness matters. It's the thinning that matters. If your retina is thinner compared to other individuals, it might be just the biological variations among the populations. So also have to take that into account. So longitudinal imaging might be important to indicate the change.
Oh, interesting. Yeah, so it's about your individual thinning rather than, a population level thinning. And also it's that question of causality that we see with so many risk factors. Is this causing it, or is this a really early change?
Exactly.
Fascinating. And so about that too, we have somebody who's also wondering here about something, who's sharing an experience where their husband, who has Alzheimer's disease, has been experiencing lots of visual issues. And I believe that she's asking about whether these pathological changes that you're seeing in the eye can actually lead to more experiential visual changes in Alzheimer's disease.
That's a great question.
Yeah.
So it depends, I would say. So there has been studies showing the correlation or connections between the different visual function disparities or visual function reductions to the Alzheimer's disease. But sometimes depending on what's the site that the Alzheimer's related pathology happened in the brains. And a lot of times when for cases for some patients, the neurodegeneration happened in the areas around the visual pathways or around the visual cortex, and that will indicate that will manifest the visual disparities happening there. And also, but that is not really entirely related to retinal change. It's simply because the neurodegeneration happened in the visual cortex itself. And there are other cases where for example, there's correlations between the pathologies happening because of the tau or the amyloid beta, and there's a correlation to the retinal function change. For example, there's a lot of cognitive tests showing their correlations. So that there's definitely correlation there, but the mechanism can either be happens in the retina or in the optical nerve, as we shown the shrinkage of volume or the visual cortex. So which side it is, we need to look at them in more details.
Right, of course, and I think it's hard for some of us to remember who don't have a background in this, that there's vision isn't just happening in the eye, but it's also happening in the brain and the visual cortex.
Right, yes. So that makes it more complicated.
And I was wondering about following up on that. I know that there's different forms of dementia, particularly with Alzheimer's pathology, such as posterior cortical atrophy, where visual symptoms are actually predominant. And I was wondering if your team has looked into that at all, if there's any literature about it, if in those forms of dementia where the visual symptoms are predominant, are you expecting or have you seen more of this pathology in the eye, or is it unrelated to those symptoms?
Right. I guess that's related to the question of differential diagnosis to see to see whether so which side of the pathology is connected more. So we haven't looked specifically into differentiating different type of dementia or different type of even Alzheimer's to look at their level of effects on the retinas. So that's a great question. That's further fine-grained detail to basically give a different level of the disease. I have to say we are not into that stage yet. Yeah. At this stage, I have to say the pathologies in retina is actually much less significant compared to the brains if it ever presents. Yeah. So there was even a debate about whether it's some pathologies happen in the retina, and then so we need to basically we need to find a better way to detect those subtle changes first, and then to give them different levels in the next stage. So, but that's really important next steps.
Interesting, yeah. And we have another question here who's asking about kind of, I guess, a symptomatic expression or maybe connection here. Noting that they often see quite vague facial expressions in people who are living with Alzheimer's disease, and sometimes impaired visual expression. And they're wondering if that also might be connected to some of the things you've talked about today.
So I guess that's a more complex problem. And so if the facial expression is also connected to the muscular functions in the brains, and so what we are specifically looking at is the visual function. Yeah. And I would say there might be connections in the brains where different part of the brains are actually affecting at different stages. So there might be pathological cascades happening between different stages. But in terms of the connection to the retina, I'm not. I can't think of at least I'm not aware of the connections between the visual functions to other muscular-based things, for example, facial representation.
Yeah, of course. I mean, I think it was really interesting the way that you noted there, especially at the end too, that there are really many different pathologies that we're looking at here too. Instead of, of course, there's a lot of focus on the protein expression and aggregation, but also these vascular changes earlier are so important. Which I think is really interesting. And I think probably a lot of people listening here today were very taken and very interested by the idea that the optic nerve and the retinas in the eyes are also part of the brain and do have those connections. And perhaps I was thinking this question might be thinking about other cranial nerves that also perhaps connect to the face and wondering about whether we might see pathology there. And it's a very speculative one, but if you could see it in the optic nerve, if you could see it other places as well.
Right. Yeah, I think those questions are interesting question linking to the systematic medicine. So if we want to look at connections, there might be connections both in the nervous system or central nervous system or the vascular system. The reason we are interested in the vascular system is because it's the updated vascular two-hit vascular hypothesis that presented, and sort of the vascular is have been considered as the root of a lot of diseases. And so those are the things we're focusing on for the nervous systems. And there's definitely connections there because they are also ubiquitous within the body, both the central and peripheral nervous systems. In the current sort of theories, the nerve, so the disruptions in the nerve is happened as a downstream compared to this vasculature. So I guess that's something important things to notice about the pathological cascade.
Yeah, no, absolutely. That's so interesting. And I think coming off of everything you've spoken about here about the pathological cascade here, somebody is wondering about, any treatments to kind of protect or stop these changes, and about whether they exist now or maybe what they might be in the future. Thinking about your work.
Right. Yeah, I think that's really think ahead about a kind of question. So for the moment, what we are talking at is early diagnosis, and what we are hoping for is that it can also be a sort of a treatment monitoring facility, so that later on, if we have a treatment in place, we can actually see the sort of recoveries. Because the reason we want to look at the retinal imaging is it's much accessible and cost effective. So both in terms of the the diagnosis and recovery monitoring, it should also be helpful. And so actually, for example, in terms of treatment itself, for example, for curcumin, it's a compound that we use to bind the proteins that we want to detect. However, curcumin has also been reported as a sort of a protective agent that can help for neuroprotective agents. So that's something interesting to see, but that's not specifically looking protecting the dementia or Alzheimer's per se, but it's protecting the neurons as a whole. So we haven't looked at the treatments. For another reason is because most of treatments are focusing on the brain side. Because if we make the treatment, we want to make sure that our cognition functions or the neurological functions are recovered in the brains. I think that's the first target sites, and besides the retinas. And so that's the current state of the art.
Yeah, of course. I think it's so important to remember that it's you're looking at a bit of a downstream or a further away effect from the site of the main pathology. And I think what's so interesting is that you are highlighting here too with the vascular pathology and also the protein pathology was making me think of the really great work that's been going on in risk reduction these days and about trying to target those factors really early to reduce the risk of pathology later on, which again I think is why it's so important when you're talking about early diagnosis and effective diagnosis and cost-effective diagnosis. Because perhaps identifying people earlier on in the journey can help them take steps to preserve or slow the pathology that we're seeing there with some of the strategies, which I just think is just so valuable and so important, especially with diagnosis, which we know is so challenging to get in so many ways, both in terms of technology and access and that knowledge, which is so important.
Yeah, totally echo. Early diagnosis is the most important thing at the moment for Alzheimer's, I would say.
Absolutely.
Before we have the final cure. Yeah.
And hopefully it will help us get there and help us make it easier and more accessible to more people. All right, Caitlin, I think those are all our questions that we have for today, so I can pass it back over to you.
Wonderful. Thank you, Daisy. It was a great job really engaging the audience there. And Dr. Ma, you provided some excellent answers. I think, one of the most important things you've demonstrated here is that importance of early diagnosis. At the end of the day, that really is something that people are sometimes afraid of or just don't know a lot about. So this work really sheds an important light on the importance of diagnosis and the different ways that people may be able to receive a diagnosis in the future. And I think just to wrap up, my last question, it's a bit more general or broad, and you may have already answered this, but what would you like the audience members to remember? What was the key finding you'd like people to take away from today's presentation?
Yep, thanks for the question, Katie. I think the take-home message I would like to say is that it's actually a little bit beyond the main focus, is that we want to look at, if we want to cure or understand the Alzheimer's, we want to look at it as in a big picture. So we want to look at the multifactorial part. So either it's a multi-organ, or it's a multiple pathological origins. And for example, recently, if we want to look at the recently approved treatment for Alzheimer's, which is only targeting at amyloid beta, what I want the audience to take home is that we want to look beyond that and looking at the tau and amyloid together, but also looking at the different factors like inflammations, vasculatures, and those things are actually helping us to understand better of the disease and yeah, in the early stage.
That's great, thank you. Yeah, I think that's a very key piece people can take away. And I also just wanted to highlight before wrapping up that it's very inspirational to see such a large team of researchers working on this work on this project, and seeing that collaboration amongst you and your team. It's very inspiring, and it's really innovative ideas and work you're putting forward. So thank you again for highlighting that for us today.
Thank you, Katie, and thank you, Daisy, both for the engaging conversations. Really enjoyed it.
You're most welcome. So I think I'll just wrap up today's webinar, just introducing our upcoming presentation that will be taking place on September 17th. This presentation will be led by Dr. Matthew Parsons. Dr. Parsons is a biomedical new investigator grant recipient, and his project will be focusing on strengthening brain cells and determining the failure of brain cell communication in Alzheimer's disease. So again, this presentation will be taking place on September 17th at 12 noon Eastern Standard Time, and the registration link for this presentation is provided below on the slide. And I think that will conclude today's presentation. Thank you all for attending. Thank you for your questions and your comments. Thank you to Daisy for moderating the question and answer period, and thank you to Dr. Ma for his engaging presentation and his thorough answers to everyone's questions. Thank you very much.
Thank you, Katie and Daisy, and thank you everyone
Goodbye.
for participating. Bye.